Morning. So the POnTE project has been funded by the Horizon 2020 program, and it is a project that started four years ago. It is a 4-year project, so it will end this week, tomorrow, indeed. And it is quite a large consortium, including 25 different partners, and with belonging to 13 different countries. And as you can see, not only European, there are also 3 countries that are third country, including Costa Rica and Serbia. It is a project that was targeted to different pathosystems, because the call at that time was not for one specific pathogen, and 3 different pathosystems that are the Xylella fastidiosa, that having the major weight in the project, but also Candidatus liberibacter solanacearum, and some pathogens of diseases causing disease in forestry. And so here you can see the website of the project, where you can get more information if you are interested on, especially on the other 2 targeted pathosystems. And of course, being not specific for Xylella, but being the project identifies to start the research on Xylella fastidiosa when Xylella appeared for the first time in an epidemic way in Europe. Later on, 1 year later, it has been accompanied by the start of another project that is fully dedicated to Xylella fastidiosa, that is the XF actors, to which POnTE project is complementary by the call. And that's why the end of the project this week will not mean the end of the research, but the research will continue with the XF actors, the other project that is coordinated by Maria Saponari. And this was the scenario at the time of the start of the project 4 years ago, when there was the large Xylella outbreak linked to a novel severe olive disease in Apulia, in southern Italy. Later on, we know that other major epidemic appeared in Europe, like here where we are now, or in Spain mostly. And at that time, there were a lot of questions addressed to first of all, at that time, there was a need to prove that Xylella was to be the causal agent of the disease, and also to identify the local and the European and the vectors present at the local level in Italy, but also in other places in Europe, to understand what there was need to understand how it spreads in olive groves, and how to improve the surveillance at European level, and which areas are under threat, and how to control. And how to control the bacteria in the plant, and so on. So what happened? Sorry. I don't know what's going on. So let me try. How to control the vectors, and how to control the bacterium in the plants, how to control the vectors, and how to enhance responsiveness and awareness. So for the first question has been addressed immediately and was clarified in the 2 years ago when it has been proved that symptoms can be caused only by inoculation of the Xylella fastidiosa genotype that is present in Apulia. And the impact of these results is that now we have a tool to screen cultivar susceptibility and to investigate host pathogen interaction. And this now is giving to the other project, to the complementary project, the possibility to make the large greenhouse testing that has been started a couple of years ago, and extensive transcriptomic analysis, the identification of putative differentially expressed genes linked to the host response. For this, later on this morning, Pasquale Saldarelli will give an oral presentation. And concerning the local and European vectors, those are the first vectors identified in Europe. All are so far spittlebug, and it has been determined which the host preference versus negative selected host plants. So now we have an idea about this, and it has been also clarified the phenology of this vector, and some very quite interesting experiments have been done to get data with the mark recapture methodology to better understand, to improve the knowledge on the movement of these vectors under natural conditions. There are several oral presentations and a couple of posters that are and will be presented during the conference. And which is the impact of this result? That targeted survey, now there are targeted survey in Europe for the spittlebug, since they have been identified as the major vectors. But also, this allowed also trials for the control of the vector population. There will be a presentation, Enzo Don Giovanni, and experimental-based parameters to develop models for short-range spread, as it will be explained during the conference by Gianni Giglioli, thanks to the data acquired in this task. And giving also a contribution, because there has been also an important collaboration with EFSA that for the update of the pest risk assessment of Xylella that has been released this year, the data produced by POnTE has been also useful to support this work, this study. So next step that will be done in X Factor now is to continue to study the transmission characteristics. Bodino will give an oral presentation, and some insight into the transmission dynamics that we will learn some about by Daniele Cornara during the conference. How to spread in olives. And well, now it has been demonstrated that a study made by the team of Juan Antonio Navas, that there are clearly aggregate patterns of spread, and the infected olives are the main sources of inoculum. I'm talking always, mostly, on the epidemic that we have in Apulia. That was one that inspired the start of this project. These results are now in a deliverable that has been developed, has been produced, and coordinated by Juan Antonio Navas. All the results of the project will be reported in the deliverable that will be all public within a couple of months, 2-3 months. And how to improve the surveillance at European level was another question. There are two major tasks that has been carried out during the project. One, there have been an important effort of validation and harmonization of the diagnostic protocols. Francois Polyakov will give one presentation on this. And a lot of work has been done to improve the tools related to the remote sensing of the bacterium. And the most significant result is reported in this publication, in this paper that was published last year in Nature Plant by Pablo Zarco. And as impact of these results, now for the POnTE project, so this work gave a major contributed significantly to the 3 revisions of the EPO diagnostic standard that has been published by the EPO bulletin, and so that now are the EPO protocol for the diagnosis of Xylella fastidiosa. And the next step, the work that is underway now in the XF actors, is that a similar work that has been done on of the airborne campaign done in Apulia on olive is now underway in the area of Alicante, targeting in this case the almond to see or to find the right protocol to get the early detection of bacteria in almond population. Which area are under treat? So there is also several colleagues worked on this topic, and the 2 major tasks have been to determine the potential distribution under current and future climate change scenarios, and to verify, so to compare and to select the most appropriate modeling methods. The deliverable has been coordinated by Juan Antonio Navas again. Here, just as example, you can see the area of the estimated climatic suitability map for Xylella currently, and this is the evolution in 40 years according to the estimate of the change of the climate changes. And as impact of this result, there has been also the scientists of the project contributed greatly to the development, collaborate with the EFSA to develop the specific chapter in the pest risk assessment updated this year. How to control the bacterium in the plants? So some lines have been followed. Confirmation that phenotypic differences in olive cultivars are supported by different transcriptomic profile. Genes involved are consistent with previous studies in citrus and grapes. And there will be later on the oral presentation given by Pasquale Saldarelli this morning. Also, there is a search, as consequence, it has been developed a search of resistant or tolerant olive cultivars. And myself, I will give an oral presentation tomorrow afternoon. But also, it has been done the testing of the NAC and acetylcysteine applications. There is an oral presentation that will be given by Alexandra De Souza, but there is also a poster of Enza Don Giovanni in the poster session. And in POnTE has been done the first description of the microbiome in olive xylem tissues that allowed the next step to be developed in XF actor, where with the further investigation on the mechanisms of resistance in olive and on the search of resistant olive germplasm, and extensive investigation on the microbiome in resistant and susceptible olive cultivars. There will be a couple of oral presentations given by Paolo Battista and Massimiliano Morelli. How to control the vectors. A large activity has been also done, especially in Puglia, on this topic. For instance, several molecules of insecticides have been tested to identify those that are more that compatible with the legislation, are also more suitable for an effective control of the vectors. But also interesting results have been obtained with several experimental field trials concerning the management of the vegetation of the crops. In particular relevant has been to prove as the tillage, the working of the soil at a certain stage of the year, at a certain time of the year, may reduce the population of the vector, as well as the cultivation of species, of plant species that are not preferred by, are not selected by the vectors. This there will be one oral presentation, I think, given by Enzo Don Giovanni on this topic. Next step will be, as exploitation of the results, the use of vibrations to manipulate the behavior of the spittlebug. An oral presentation will be given by Avosani, and testing trapping for monitoring the population. Also, there is a presentation that will be given by Enzo Don Giovanni, and also testing other bioinsecticides, mostly fungal and entomoparasites. So, how to enhance responsiveness and awareness? There is also an important activity done, a huge activity done on this way, because just to tell you, there have been several courses that has been developed to train people involved in the laboratory for the analysis, for the diagnosis of Xylella fastidiosa, but a lot of activity of dissemination has been done on both on the social, on collaboration with the media, and a lot of public meeting or seminars have been given also in the territories affected by the epidemics. I think that there are hundreds of meetings that has been, or seminars that have been given by scientists of the project. And of course, I apologize because not all the participants are included in this picture, but of course, as coordinator, I have to thank all the participants to the project. At the beginning, 120 scientists, but on the way of the project, this group, this number has been increased, and I do not have the exact inventory of them. And since there is still one, well, maybe I hope there is no audio, but I would like just to show you one short video. Okay, that has been launched yesterday to promote the results of the project that will be soon public. As I told you, it's not only Xylella, the project. And that's it for my presentation. Thank you.